Well, thank you, Anton. And today I'd like to provide you the researcher point of view, a researcher in terms of the system biologist using all the artificial intelligence and publication as a tools to propel the biology research. At the same time, as an AI researcher, to try to get a maximum out from the publication and data resources we can access. So like title. It is a kind of new grand challenge I'm proposing and actually get started, which is to create an AI system, which I think we're going to that page to build an AI system that can make a major scientific discovery worth Nobel Prize and probably actually win it. And the reason why I call it I have like a few papers if you are interested, but like it's kind of interesting. I say like when I say like some of the discovery that win a Nobel Prize, like someone when I give a talk, someone come and said, I say, well, Nobel's will, it says they're given to the person, so it's not given to the AI system. But if you think a little bit deeper, challenge can be rephrased, phrased into like this, like well, AI system will be highly autonomous and then discover something major, and the Nobel Prize committee will give an award with noticing that it's the AI system, not a human scientist. So it's a kind of a Turing challenge at the very highest level. So I'm saying this is like a Nobel Turing challenge. Okay, so you might ask like, well, that is probably not possible because it's obvious like it's AI or not, but that's not things that not simple. And like who invented like a Bitcoin, like or the cryptocurrency. And, this single paper in a blog post of the guy named Satoshi Nakamoto. I mean, we can very clearly identify like this is really the origin of the whole thing. So like if a Bitcoin and cryptocurrency blockchain changed all the economical system, and the Nobel Prize committee for the economics have to give an award to that. And it's, very clear, have to give award to the Satoshi Nakamoto. The problem is we don't know who the Satoshi Nakamoto actually is. Whether this is obviously a Japanese name, but whether it's a Japanese or a single individual, a group of people, we don't know. Okay, so but still, like even if people don't meet this specific individual, people just like go for like Bitcoin, cryptocurrency, blockchain, simply because like what even did in this paper, well, it's posted in a blockchain, in a blog post. It's not even in archive or not even peer-reviewed journal. It is the origin of everything in this. So like why don't we imagine like if you got an AI system, you create a hypothesis, run an experiment, and write a paper, which is possible, and then put into the blog post. And if that discovery is completely really decisive, really change, then we have to recognize it. Okay, so of course, at this moment, like, whether you're getting Nobel Prize in reality or not, it's not really the issue. I mean, of course, I'm talking about Nobel Prize as a very symbolic, and I set the bar very high. That's what we're talking about. But, like, in that stage, like, a machine will probably make a major discovery, like, every 40 hours. Like, the way we do science will be dramatically changing. Okay. So that's my goal, my current research goal. Okay. So to do this, I have to do two things. One is build AI system, and you have to do the experiment. So you have to, that includes the robotic system as well. That can make major scientific discovery. Okay. And the second, it must have like a very strategic choice of what to investigate, and then search for the literature or communicating from publications, and then possibly they explain the value of the discovery or value of the research. Okay. So first one is just a discovery. Second one is the more autonomy sides. So that so I'm not going to too much in the detail, but like my recognition is scientific discovery is at the pre-industrial revolution level. We know we have like omics, the genome sequencers on the proteomics and the genomics, and there are quite a lot of like machines we can measure a lot, and then they come up with a lot of data, and they have like information processing, bioinformatics tools as well. But at the same time, like if you look at the scientific discovery process, at the end of the day, the researchers are scratching their head and they think, wow, oh my God, I mean, what is this, or what does this mean? Okay, and then people complain about like they are flooded by the data. Simply we can't really handle that data. So like it's just like this is a textile factory, I think a photo in Cambodia, and then with like all the handcrafted one, and at the end of the corner, you have like a machinery which actually spit out the all the textile, but then you have to really do the handcrafted. That's like a scientific practice today. Okay, and we're talking about serendipity by accident, scientific intuitions. So what is that? This is not well-defined terms. So like, one of the purpose of this research project is really to have like a well-defined understanding what serendipity or scientific intuition, or possibly redefine them, or probably alternative form of the scientific discovery. Okay. And of course, like, I proposed like system biology 20 plus years ago. And after, spending a lot of time, I'm an editor-in-chief for the one of the Nature partner journal, System Biology. And if you look at, all the publications, we can handle like a very precise model if the scale is small. Okay. If the scale is big, we tend to use like a rather statistical analysis rather than a very specific simulations, simply because we can't really handle and digest the knowledge of that scale and do the proper like a parameter tuning or proper analysis on all the details. So we have to go for the statistics in that case rather than the mechanism-based simulation. So so one of the things is that we can't really go for all the details and we can't really handle that. So system biology is the science for sort of AI or AI-human hybrid systems. That's the conclusion after 20 years of system biology. And then as interesting with this is we have a few cognitive issues. Okay, there are some other issues as well, but I'll focus on cognitive. One is the information horizon problem, information gap problems. I'll explain some of them. Information horizon, this is actually statistics I think like it was 10 years ago. In the biomedical field, 1.5 million papers published per year, which is like a 4,000 papers per day. I think we have now 2 million papers per year in the biomedical field and 5,000 papers plus per day. And if you select the area like neuroscience, oncology, or immunology, I think hundreds of papers per day, but simply you cannot read it. So we're doing the research with ignoring most of the publications, and we just sort of randomly pick. You might pick Nature and Science, but of course there are important discovery in the minor journals as well. And also you want to integrate the knowledge. This is cell signaling. We read 1,500 papers, not just abstract, all details, material method, and I got high quality curation. Spend like a few months to get into this. Is like a most comprehensive signaling network diagram on the eukaryotic cell, and it covers everything basically. Now this is like an e-cell cycle. I think we got about to almost 2,000 papers, most detailed description of the cell cycle of any organs. Okay. And this is the immune to like receptor pathway, and this is like Alzheimer related. So the issue is like we've been doing it manually. Okay, we read the papers, we download the publications, read it very carefully, double checking it, and you gotta post it and you wrote down, and then got in the computer graphics. Okay, now, so this has been the gold standard. High quality than any of the database, because like we have all the mental model in the team, so we know if there's a mistake, we immediately understand what's wrong with them. The problem is we can't keep doing this. Okay, so like we built this like 7 years ago, 10 years ago. We can't update. We can't do it because it's psychologically impossible for anyone to actually keep working on just reading a paper, writing descriptions. And then or the society, what is the career path of the people who devoted for that? Okay, a few months, fine, and they are very knowledgeable and do the experiments, do the research. Those experience and knowledge helps. But you can't really have assigned people to keep doing that for the rest of the life. Okay. Now, second thing is the minority people. If you have like 90% of papers talking about A activating B, 1% talking about the other thing. So like a people do we notice that? Okay. If we notice that, do we dismiss this as a mistake? Or like you think like this is a new discovery under specific conditions, and you have to deep dive on that. And we're not really good at identifying those and going for like if there is one case, we can do it. Probably 2-3 cases, you can do it. But if you have 50 of those cases, we can't really do that. We just ignore that. That's the reality. Now, also we think and reason and communicate in the language, and the language has cultural context and all kind of cognitive bias. And then that became problem in the clinical reasoning. And of course, that became the problem in the understanding, describing, and communicating with the peers. And that impacts our mental model. For example, some of the genes called the oncogenes. Oncogenes is a very highly contextual name because that implies the functions, and it was discovered in a way specific condition that genes seems to act as a promoting, the cancer. Later days, some of the oncogene found to be cancer suppressor gene, but still, so sometimes it's conflicting, because initial context of discovery labels something, and that semantics has its own life. Okay, so those things actually is really inherent in the our daily practice, which we depends on the language in the reasoning process and the communication, and that actually is a limit of human cognition. So one of the way to solve or mitigate the problem is use the power of computing and the power of AI. And if you look at the AlphaGo example, and which is very easy to understand the example. Now, what they have done in DeepMind is you got all the record of the Go play, and then put in a database, and then using deep learning network to be able to predict what's the most possible next move. Okay. And not only that, they actually play against the AlphaGo against each other, and then with the Markov random tree search. So they have some possible likely move, and then some randomness. So there are many cases, and then play against each other, and then against so they go to traverse into the massive tree search, and then on each step they evaluate the likelihood of winning or losing. So they evaluate, and then get the reinforcement learning to be able to generate the better move. Okay. So what is this? I mean, this is basically if you think this is the outer diagram to be the all possible move of the Go, and which should be gigantic, and then this red circle in the middle is all the recorded history of the all the motions movement of the Go. Okay, so which is the Kifu in the Go, and which is probably a very small fractions. And then, so what AlphaGo did is memorize this. What is this? Learn from the movement, and they get the most likely move, and then I got the the Markov tree search and the reinforcement engine. So they actually expand the search space horizon at the vicinity of the historical play. So a possible move that past, that can be inferred from the past human Go play. Okay. So by the time AlphaGo play against a human Go champions, AlphaGo has already experienced a much bigger search space and already evaluate. They knew what's going to happen. AlphaGo knew what's going to happen, where the human hasn't been exposed that at all. So no wonder why AlphaGo is strong. Okay, so then actually move on to the AlphaGo 0, which is very interesting. They get rid of all the records. So they no data. Okay, people talk about big data, but AlphaGo 0 is no data. So it's randomly make move and then evaluate, and then because it's computational intensive and the massive search has been done, unbiased search. So basically what happened is the AlphaGo not 0, AlphaGo start from what the human have done. AlphaGo 0 at unbiased search of the entire Go space randomly, and then the result is AlphaGo 0 is way stronger than AlphaGo. So that means 3,000, 4,000 years of history of Go. What we have established is the kind of game strategy which humans likely to make a move or human likely to understand. It's not necessarily the strongest move of the Go. So that's interesting because we are biased. Okay. Well, can we apply this kind of strategy to the scientific discovery? So that means what we create an engine for scientific discovery. Okay. Scientific discovery in AI is nothing new. I mean, it comes from the immediate after the birth of the concept of artificial intelligence. Joshua Lederberg, Bruce Buchanan, and Feigenbaum, with the dendron and metadendron, for the mass spec and try to infer what the molecules are and trying to discover what's new. And then the recent, and then the many projects like that. And then a recent one, for example, Ross King in Manchester, now in the Chalmers in the Gothenburg in Sweden, create the robot scientists which can generate the relatively simple hypothesis on each genetics and run the experiment and verify. So it's a closed loop system. Okay, so this is interesting. So like a little bit history. So what we're going to happen with the endeavor is defining a scientific discovery. And then basically my hypothesis in the project is a massive search and verification in the hyperspace in extreme speed. And so we're going to scale the discovery. Okay, so if you look at the actual major discovery, for example, like IPS or conducting polymer, what happened is in IPS case, like Professor Yamanaka going to the phantom database and identify 24 genes which can possibly reprogram the cell and gain the stimulus pluripotency. And then from there, he got the optimization. He just did leave one out. I mean, his student Takahashi-san did a leave one out experiment and identified a minimum viable set, which is like a Yamanaka factor, which is 4 genes. Okay, so this is basically the search and optimization. And Shirakawa sensei's the conducting polymer was interesting. They got got the intern from the Korean Electric Corporation come to his lab and try to understand polymerization. And the student got experimental condition completely wrong. Like 100,000 times actually much more dense liquid agent was poured into the experimental system, and then accidentally got the thin film. And then Shirakawa says, understand, oh, this is interesting, and then optimize the conditions. And so the polyacid thin film formation was established. And then with McDermott and Heger, they come up with like a way to optimize the conducting polymer. So this leads to the Nobel Prize in Chemistry 2000. So this is basically accident is search space expansion into the extremity, and then optimization of the condition, and another search and optimizations result in the conducting polymer. So like basically many of the biological and chemical discoveries and probably others is a search and optimization process. If that is the case, like how can you do that in a computationally? I need a platform, so I keep building it, and then before trying to create like a really the system which can do that, we have to connect the lab. We have to because like an experiment, we need to tap into the old experimental devices and a data resource and all the things. So like our lab has to be fully connected, and then they put the AI assistant in specific cases, and you get the autonomy and you're able to fully control it. So like, and then we've been building platform like a Garda platform, Gandara platform. I'm not going to talk too much in detail on that. All the connectivity established on the wearables, and some of the project was actually I just go back. This is like a JSPS project on the where the Professor Kurachi and Osaka University was head, and then I was like kind of sort of co-chair kind of member. Create like 6-7 software module connected to each other, identify the prediction of the drug-induced arrhythmia. Okay, so this has been software side. It's all connectivity, not much on the AI yet. And then connection with the Shimazu, mass spec, and other thing as well. And then one of the example of how you put this connectivity with the AI is with the Professor Kawaoka on the avian flu drug discovery project. And you have like some pipeline drug discovery. One of the things we want to do is we want to understand what is the molecular interaction for the virus replication cycle. Okay, so what we did is again go back to the papers, where there's 7,000 papers, and I get the virus influenza infection duplication network. Oh, by the way, this was in the basis for the international project in the corona virus replication, drug discovery project, because like this is the virus replication project, not just influenza, but like all the virus. So now, at that time, like initially we got it like a hand curation, but now we can automate it. And not entirely, but like a highly automated. We get deep parsing, natural language processing to be able to digest 1,000 of paper, get all the relations extracted, and then several approaches with the deep learning and all other. We come to the stage that we can reproduce a molecular injection precision. So like this diagram indicates there was a coverage. So like a white one's the baseline is a hand-curated one. This is the gold standard, and then the green one is automatically reproduced like a molecular interaction. So we can now reproduce 95 to 97% of the what has discovered in a human, and can be properly reproduced. So and then what happened is 3% is we have to still tune. And that's interesting is something human haven't discovered. has been suggested by the AI system. I think we're going to get to that at the end of the talk, but like what we found out, that is actually usually thought like that's a false positive. That turned out most of them is not false positive, because like those relationship which was not really curated in human was actually discovered and confirmed in the publication database. So like we come to the stage that some of the machine we have, AI system, will be able to not only reproduce, extract the knowledge, but also predict what may be discovered in the future. Okay, and then have like some kind of a model. Now also like and this is kind of look like a gene expression dendrogram, but this is actually for the older corpus. So like this is what kind of terms has been used in documents, and then depends on the document talking about autism, depression, schizophrenia, and then you have like this kind of analysis. So this is enormously useful for the drug discovery. And then with that we can get a network. And then we use this network, which is extraction from the publications, to do the control theory understanding, and then we'll be able to, get to the, control theory to understand what is the possible node which we should be control to be able to stop the virus replication, right? So this is not really the AI, but like a control theory type of. Oops, it's not moving. Okay, more rigid mathematical control. Okay, now, so we identify mathematically what is the possible target, but you have to go back to the data as well, and get all with the omics data. What we measure is the interaction, protein-protein interaction between the virus-associated proteins and proteins in the this is a virus replication cycle, which is associated with a specific target process. Okay, virus replication cycle can partition into the basically 6 subcomponents, sub-process, which is virus entry, and then disassemble, replication, deassemble, and exit, and things like that. So we have like a 6 box on the top, and that is a sub-process. Okay, and in the circle on the bottom is a virus-associated protein. This is influenza. So what we want to do is cut the link in between them, and you can't cut the link everything because that will kill the cell and that will be toxic. So we like to identify what the molecules are in between. Okay, so we have like this purple dot in the middle, and those are the things like which wants to target. At the same time, some of the target may not work and then have not much effect. So we combine with the control theory analysis, and then we identify what are the real high priority target, and then with that. And then library chemical, we do the docking simulation, which is basically I just skipped all the technical details. All the proteins structure in the pathway in this network against all the possible compound, which this is a part of that, and you get heat map. And from this, not just compound target, but also the collateral interactions. So like what is the network effect? Which compound will have like a maximum network effect, specifically stop the cycle, and then a minimum side effect. Okay, so that we're going to compute. And then also the protein structure we're extracting not for the database as well, but like publication trying to look into that as well. So there will be a text mining component to that. So although we have a long way to go to the computer automation. So this is actually an example, and then of course we come up with like 3 prime candidate and doing very well. This is the example how the publications in a natural language and then combined with the database and the figure can be used using AI to reproduce the core model, and then with omics data we'll be able to tune down prioritize a possible target, and then what are the lead. Okay, so this is interesting. So the question is, can we do that in a scale? Can we do it in a scale? Okay, so this is a diagram which actually shows a protein-protein interaction in human. Okay, and this is in the right side, and this is very consistent with the human, mouse, and then the Drosophila elegans. Mathematical structure is all the same basically. And what's interesting is to the left side is that K is a number of known interaction for the molecule, and then frequency is a likelihood that like a molecule in that category to be drug target. Okay, so K50 or 100, this is like a lot of interaction. That means that this is a very important molecule, like a P53, things like that. So like a lot of papers and a lot of curated. The problem is it's unlikely to be druggable, because if you touch that, that has an enormous impact to the cellular system, and it's likely to have a lot of side effects. So most of the protein on that category is targeted only for the cancer drug, because cancer drug, you have to accept some kind of a toxicity to say kill the cancer. So that's it. So like an interesting one, the middle side is actually very efficient, highly likely. So this is talking about all the backbone. Okay, so broad effect, but less toxic than the all the highly connected genes. But the interesting part you should notice is the one on the left, and that is like a K1234. It is a molecule which is not really interacting much and less investigated, but has a reasonable chance of being a drug target. And there's tons of them. Okay, so like each probability of getting the drug target is relatively small, but like there's a lot of them. Okay, so this is a long tail part. But the question is like, how are you going to go for it? Because if you assign postdoc, okay, please do this. This is one of the molecule, and likely this guy is successful is very low. Okay, but if you do it in a number, most likely that you're going to get something. So like, for this kind of thing, you have to automate it. You can't really do it in a human hand. So we're gonna have like an open platform of collaboration to be able to do that. And to facilitate this whole process, we build like a Tuxedo, which is like a text-to-knowledge system. So we have like a lot of applications like hypothesis generations and evidence and drug repositioning, all that. I'm going into detail. But like basically, Tuxedo access to all the open source, open access papers and full text and PubMed, and we have like a 20 million articles in PubMed articles, and whenever it's available, and then in our database, download it, and then we actually compile. We go to work to make a deep learning-based analysis and a deep parsing as well. So gigantic database we build it, and then from there we got like all the relationship. This is a galaxy. What we say it's like each dot is a paper. Okay, and then pain. This is the one of the center of gravitation of the paper talking about the pain in Alzheimer context. So like this is a galaxy of the paper talking about Alzheimer disease, and the pain is actually center of the gravity, and this is a cell organized map and amyloid and then all that, right? So we can actually dynamically curate it. We're going to detail, and this is another view. This is the 2D view. And then also we can have like if you have a patient in the middle, a patient, and what are the possible like rewards in the preceding or before after the patient. I have like tons of them. We can do for like 20 million papers, right? Okay, so we can do a lot of analysis on that. So that recently our team come up with like deep learning based method to process over those database. And then I come up with the very interesting prediction. So when you have like a one term, for example, like proteins or disease is associated, then most likely that could be the cause of the cure for that. And then we actually partition to the all the decades, like every decade. And then what is the possibility of that association to be discovered in the next decades? So we, this is the blue one is a virology. We achieve like almost like a 93% accuracy on the prediction capability and things like that. Okay, so and this one is most interesting. Okay, so this is where you use the same algorithm for the specific cases. Okay, RDFO is Raynaud disease and fish oil. We found out Raynaud disease can be cured by using the fish oil and all that. Okay, migraine disorder for the magnesium. And discover year is RDFO. This relation discovered 1985. And then if you partition all the paper, 1970 to 1979, likelihood this association was discovered or predictable. It's not discovered because it's not there, right? So like from the paper, which is not talking about association, can we predict this association, right? So that's this number. So like this is only 4% probability, confidence that this is associated. for 90 to 97, but 80 to 89, 61%, because this is obvious because this was discovered 85. After that, there's a lot of paper talking about it. So 61% and 90 to 19 years, high associations, things like that. But what is interesting is this Y-1 column. Okay, so we took papers up until the year before the discovery, and then predict, whether this association can be predictable, year before that association was discovered. Okay, so the RDF4 is 0.6, 61%, and then magnesium is 98%. So we have quite high confidence that the association to be predicted a year before the discovery of that association. So this is very interesting. Now, but this is only association. What we're trying to do is, given the association, using all the text mining, can we actually get to the mechanism of understanding how they are connected? Then we can actually tell the difference, right? So then we're talking about some other thing. Okay, so this one is Notch and GCN2 was 3 hops away. We can now talk what the intermediate association, and then that was actually confirmed. So then direct connection was confirmed later days, things like that. So we're actually all the benchmark on this. Now, with this, we can have a lot of possible associations, possible mechanism understanding hypothesis, and then we can, plan an experiment to verify the association before anyone else notices that, and do the experiments, and then we can do in a scale. Okay, so if you talk about a biological discovery, most of the biological, not all, is about explaining the mechanism of biological phenomena, either the disease level, either the molecular understanding or specific molecular interactions, phenomena. And then basically, we want to have a possible combination of molecular mechanism and then interaction in between them, and then what are the molecular involved. And that is all what we're trying to, discover. And this will be nested most of the case, but this is the basic fundamental. Okay, and then we now come to the stage this fundamental can be discovered automatically. I mean, it's a little way to go, but I think we're getting there. So if we do this, we can do all the literature search and then unbiased search of the entire scientific literature and trying to find out what's going on. So this is a diagram we used in AlphaGo explanation. So among the scientific truths, I mean, probably this is boundaries, one. But what we already knew from the paper is the knowledge we discovered. Okay, from there we can expand the horizon to the human discovery knowledge. Okay, I think that's worth it. So we're gonna start from there. But at the same time, after that, we probably want to go to a much wilder search, which is unbiased search of all possible scientific truths, with the basis that we don't know yet. But I think we can keep going that. So, but at the same time, with that territory, machine may be able to discover the truths which human have hard time understanding it, because there is no guarantee the scientific truth is in a form that we can easily understand. Okay, but to do this, I think we need the AI, ML, and also a model simulations and high precision experiment. This is a little bit of bottleneck because you have to, you have to spend time. And we can speed up all the AI process, but experiment you have to wait. Okay, so how we can do the in terms of the virtually and then confirm a minimum number of the experiments in the physically, that's the issue. And then we're talking about all the existing data and data acquisition, and also not only the data acquisition, virtual data generation is very important. And then existing data we can probably discuss. So virtual data generation, can we actually assume the specific biomedical conditions, and can you verify that it is actually true or not? I mean, that kind of simulation. So we have to, okay. So some may actually argue important thing in scientific discovery is asking right questions. Can machine ask the right questions? Okay, asking right question is sociological phenomena. Basically, for a scientist to be successful within a specific period of time which you're active, and then given the resource, you have to ask right question to be successful. If you remove that constraint, well, we're probably not able to remove entirely, but reduce the constraint. Rather than asking that question, you ask a lot of questions. You ask every question. Important thing is in there. Okay. Of course, you can't entirely eliminate because there will be resource bottleneck where you want to know something earlier than later. So, but still, with that, AI system, we can ask more questions. So that means, we'll transform the way we do science because we ask every question. And basically, asking right question is specific value of that individual. So this really depends on scientific conditions, whether the scientific intention that person actually right or not is not clear, and right, that that intuition is correct, and after the effect. Okay, so like what we want to do is unbiased search over the search space. Okay, so basically we'll define scientific discovery. And then the interesting is, like, if you create this kind of AI agent, is it going to have one AI system do everything, or are you going to have like characteristic AI in a specific domain and then talk to each other as well? So this is interesting. Which one is more efficient? Which one goes to the grow faster? So if the multiple characteristic AI scientists right way, then you're going to see the multiple AI scientists which will crystallize the idea, take it from there, and I get to the expansion and jump into the other field, combine the connecting a dot. So it might actually look like a career path of the real scientist. Okay, where the left one universal scientist, they have a really sporadic major discovery in unrelated area randomly. So it will probably not look like a human being, because like this, a little bit like weird, right? Human has no capability of doing that, usually. Okay, so if we got through this, we'll have like a process of scientific discovery, serendipity, by accident, scientific decision. Probably we can do it, we can define it computationally. And so that will be extremely interesting. So this is really the actually a combination of the what I have been doing in the past, like a massive parallel, large scale AI and a system biology combined together, and I get to the new horizon. Okay, so the implication, this is that slide, is I think what we are trying to do is find an alternative form of scientific discovery, and possibly alternative form of the intelligence, and accessing scientific science at unprecedented speed. And probably machine might evolve by itself. But I think our basis is like not just the data, we need to have an interpretation that we have taken from there. So it would be very interesting that this AI system is deeply embedded to the publication platform. So I think we are very much interested in working with the publishers, because we want to have like access to all the informations through text. That's very important. And then take it from there and help the scientists to discover things faster. Okay, thank you very much. That's my initial presentation. I think we can go for the question and discussion period.